Hello and welcome to Naked Genetics, the show where I seek to find out just what's going on in your genes. I'm Will Tingle, and join me as we seek to cover the latest in genetics research and see what the front line of genetic mapping and sequencing can reveal about our world. In this episode of Naked Genetics, what new DNA techniques are revealing about human sacrifices in Mayan culture, we debunk the junk in junk DNA, and the upside-down sea snail that makes rafts out of its own snot. First up, let us delve into a couple of new pieces of genetics research and get our in-house genetics experts to give us their say on it. It's time to link back up with the ever-indomitable Shivani Shukla and Erwin Scally. First up this week, a genetic study has uncovered some fascinating details about the life and death of those put to ritual sacrifice in the ancient Mayan city of Chichen Itza. Modern-day Chichen Itza is located in the Yucatan Peninsula in Mexico, and in its heyday was a site of major significance for the Mayan people. At that time, ritual sacrifice was a somewhat common means of providing nourishment for their gods and goddesses, and was a lasting practice up until the late stages of the Spanish conquests in the 17th century. Now, a new study of ancient DNA remains found at this site are thought to be around 1,300 years old and have revealed more about the demographics of those selected for sacrifice, but also what happened to the genetic composition of those indigenous populations in the millennium since. Eowyn, to kick us off, given the age of these samples, how was this DNA procured?
My understanding is they were extracted in the usual way for ancient DNA, which is from, in particular, the petrous bone, which is a bone in the inner ear. It has a high concentration of DNA. It's quite a dense bone and therefore more resistant to sort of the leaching out of DNA into the environment. And I think that was what they did, because they did mention that for some of the samples, they weren't able to get that that bone wasn't found, and therefore they weren't able to get DNA.
Shivani, to bring you in, I feel like this is a bit of a research into the bleeding obvious, but how do we know these samples were as a result of human sacrifice?
The site in which these burial remains were found is just one of many Mayan sites where these human sacrifices are made. As studies progress and we understand more about the Mayan culture, we know that human sacrifices, whilst not as common as we believe, were part of their kind of giving back to the gods, and they believed in the importance of nourishment. So we do see other very similar kind of mass burial sites in South America, at least.
I don't know if it's true for all of them, but I guess the fact then that you find a large number of children all in one place, which is suggestive of that not being just a normal burial site. So I think that's some of it is inferred and due to the religious nature, and we know that ritual sacrifice was an important part of Maya culture.
And I suppose being found in a cenote, it's unlikely that they all went down there and just died by accident.
Yeah, I mean, these were all found in underground caverns. So we know, I think, that the Mayans believed there was a connection with gods in these caverns. And also, like Eowyn said, there are many similarities between the remains. So there seemed to be a preference for twins and a preference for children. And I think they were all male. That cannot be by coincidence. They're clearly selected. And twins play a really important role in Mayan culture, and one of their kind of main religious texts have this concept of hero twins who sacrifice themselves for the betterment of the world. So twins are a recurrent theme we see in many burial or sacrifice sites for the Mayans.
You do raise a good point, though, Will, in that one of the opportunities that arises when you have a new source of evidence, like genetic evidence is to keep an open mind with regard to some of the interpretations that have been made based on other evidence, and to say, well, is this also consistent with those stories? But it is also true, maybe not here, but certainly in other sites around the world, that we have discovered things through genetics that maybe don't necessarily agree with every aspect of the story that people had proposed before. So it's worth asking those basic questions.
So much like the Rapa Nui study of last month, this is a good bit of DNA confirmation of a previously posited archaeological theory.
I think in this case, yes.
As you said, it revealed that, the preference was for young males and even twins due to the ideological preference of that being a more potent sacrifice. Did it reveal anything about how they lived before they died, though?
A lot of their diets had been quite similar too, and I think the interpretation of that was that they had been eating a diet as part of a ritual in preparation for sacrifice or something. I mean, one wants to watch out with interpretations like that, because that's something where it would seem there's a number of possible explanations.
I mean, it would also make sense if children were to be living in the same area where the certain type of crop is grown, that everyone's diets would be similar. So I suppose it's hard to make the distinction of whether the diets are pre-selected for ritualistic purposes or everyone's just eating the same thing because that's what there is.
Yeah, we should maybe clarify also what we mean by isotope data. That's another kind of molecular data that one can get from ancient remains, not genetic data. Essentially, you're looking at the particular types of molecules that you find, and there are different versions of molecules that are often characteristic of different origins in the ecosystem or in diet. And so that actually does give you an insight into what people have been eating and into their environment that they've been living in. So that's been a part of archaeological science for a few decades. And so this is, it's quite nice now we get these studies that really combine sources of evidence of various different types, including genetic evidence, to really build up a picture.
I suppose a good secondary side effect of this study is that you get a genetic map of the individuals, the indigenous people that lived in that area, kind of pre and post colonization as well. Do these remains show that the modern day populations have had any significant alterations to their genomes in the time since?
Well, I think the main thing actually is that, or the first thing to note is actually there's quite a lot of similarity between the nearby modern population and the population of this ancient city, and we're talking about going back well over 1,000 years, I think about 1,200 years or something. And so there is quite a lot of similarity. So that suggests there's a lot of shared ancestry, that a lot of the ancestors of present day people are similar to the ancestors of these people in the ancient city. There were a few changes. One, for example, being that these ancient individuals seem to have a little bit more genetic similarity to present day and ancient samples from the Caribbean. So there's some shared component of ancestry there, which has since been either diluted or lost in the present day in the modern population.
I think it's interesting to note that during colonization, the Mayans were, well, not only persecuted, but underwent a lot of natural selection because they were exposed to pathogens such as smallpox. So by comparing kind of the alleles with the remains and the present day population, it does actually support that the present day Maya have undergone a large degree of pathogen selection. You can tell by the overlapping of these particular alleles. So it's another incident, I think, of what we think to be true versus what is represented genetically. I think it's just interesting that, things have advanced so much to the point they know which alleles correspond to which diseases. So, there's a specific one quoted here, which is a binder for salmonella peptides. So I think, as things become more sophisticated, not only can we say, oh, yes, there's been natural selection and pathogen resistance, we can even say what to. And I think that's really interesting.
Thank you, as always, to Shivani Shukla for that offering. And we look forward to having your expertise on the next programme. But now it's over to Aylwin for a very bullish story. A study published in Nature has appraised the genetic impact that an extinct species of cattle has on our modern populations. The auroch was a wild bovine that appeared in Europe about 650,000 years ago. It grew to a height of 1.8 metres and lived throughout much of the continent before being driven to extinction due to hunting and habitat loss in the 1700s. But their genetic legacy lives on. The analysis of ancient auroch DNA taken from a bone found in a cave in Derbyshire has revealed when and how these wild beasts were slowly converted into our more docile domesticated cattle. Aylwin, what extra dimension does this DNA bring that we didn't have from just looking at contemporary cattle DNA?
In a very straightforward sense, it's just a direct comparison. We know that modern cattle derive, and there are various versions of modern cattle too, but they all derive from domestication of ancient aurochs populations. And so you're right that we can do, and there were people did initially do studies of just looking at modern cattle and trying to tease out, can we work out what the ancestral relationships between these present organisms are. But it's just, now we've got a thing to compare with. It just makes it a lot more easy. You're trying to do a jigsaw puzzle without looking at the box. It's a lot harder when someone shows you what the picture is supposed to be. And to some extent, seeing the ancient genomes from which the present day domesticated cattle derive, that helps us solve that puzzle.
What do we now know then when we compare the two DNAs, and what insight does it give us as to when humans started to domesticate them?
But it's quite an interesting picture, actually, because today we have, broadly speaking, there are two groups. There's the Bos taurus, which European cattle are derived and which are familiar to all of us in the fields around us in Europe. And then there's also Bos indicus, which is the cattle domesticated in South Asia, and all or nearly all Asian cattle are derived from that domestication. And it's believed, I think, still that those are the two main domestication events. And both of those derive from ancient wild aurochs. There were different auroch populations around the world, just like other animals. And we can see ancient European aurochs and ancient Asian and North Asian aurochs. And this study was able to sequence DNA from various samples of all of these different auroch species. And it seems that the aurochs, their common ancestor goes back kind of couple of 100,000 years. There's a sort of divergence of the aurochs into different groups in Asia and Europe. And then subsequently, the European cattle, those emerged from the domestication of some wild aurochs population in sort of the Middle East, Southwest Asia, that part of the world, and then spread from there. As Europeans moved out of the Middle East and brought farming techniques into the rest of the world, into Europe, etc., they brought their cattle with them.
We've got the timeline then. Do we have any idea about the process itself? Because it's kind of fairly widely accepted now in the case of wolves that we managed to pick the most docile ones and kind of inadvertently selectively breed from there. Was this a similar situation?
I don't know that we know anything about how docile they were, and I suspect it's conjectured that they probably weren't. That a wild aurochs would have been quite a formidable beast, and so therefore there is some evidence from this study that actually the initial domestication event didn't involve that many animals, probably because it was it was somewhat risky business to try and go and capture one. And also, if you think about it, suppose I want to start domesticating some animal, a group of cattle. Maybe the thing I do is I somehow kind of put a fence around a whole herd, and thereafter that's my herd, and I start kind of trying to domesticate the offspring and so on. Actually, it seems like an easier approach, which is what was taken, was you just capture one female animal and you somehow tether that or keep it within your control, and then you allow that animal to breed relatively freely with the wild population, with wild males. But then you've got control over that female and her offspring, and therefore you can build up your stock that way. And it seems that's what's happened, and therefore we can see evidence that the domestication is somewhat female dominated at the earliest stages, and that there's lots of ongoing interbreeding with the wild population, and that carries on throughout the early domestication period. There's a number of these similar domestication stories where we're trying to put it together, and it's surprisingly hard. And one of the reasons why it was hard actually is that this pattern of ongoing with the wild population, it's not like we take the whole wild population, domesticate that, and then there are no more wild animals, no more wild version. The wild animal continues to exist, and this pattern seems to be something that's actually the nature of domestication, that it's not often. In fact, I'm not sure that we've found any examples where there was this sudden domestication and a complete isolation of the domesticated crop or animal.
Erwin Scally, thank you very much indeed.
Music in the programme is sponsored by Epidemic Sound, perfect music for audio and video productions.
You're listening to Naked Genetics, the show where we look at genes and the geniuses that study them. And for the most part on this programme, we look at DNA that codes for stuff. The bit of our genome that creates real tangible proteins or what have you, that go on to change our body in some way. But do you know what percentage of our entire genome, everything stored in our DNA, actually directly codes for stuff? It's about 1%. So what on earth is the other 99% doing? Well, for a long time we had a simple answer. Nothing. It was junk DNA, remnants of bygone protein recipes that we no longer have any need for, or stuff that came in via disease or other means that we have nothing to do with. And for many decades this opinion persisted. But now we realise that a lot of what goes on behind the scenes in our DNA is heavily controlled and moderated by non-coding DNA. So today, let's have a brief peek behind that curtain and debunk the junk. Starting with the idea that certain non-coding DNA is responsible for part of the splicing process of specific genes. Say you want to take one recipe out of a cookbook, you need to know where to start and where to end looking at that recipe, so you don't accidentally add a cake ingredient to a pasta. The presence of this non-coding DNA as a means of selecting the right bit of genome to make into a protein can mean very rare health problems present themselves if this process goes wrong. I've been speaking to Mount Sinai's Ernest Turow, author of a new publication on just such a disease. And he began by giving me a rundown on how attitudes toward junk DNA have had such a pivot in recent years.
The term junk DNA was coined over 50 years ago by an evolutionary biologist, the same year that the term junk food was popularized by a nutritionist. And just as junk food had no nutritional value, junk DNA was used to refer to regions of the genome without a known function. At the time, that would have encompassed almost the entire genome. But over the years, it's become apparent that actually quite a lot of the genome is functional. About 10 years ago, colleagues in Cambridge published a paper pointing to a very large proportion of the genome being, biochemically active. I think the figure was 80%. And that statement actually stirred up a bit of controversy at the time. And I think there's no real consensus on what exactly should be considered junk DNA. So I think the term has fallen considerably out of favor in recent years. I think it might be more useful to think of the genome as containing a vast array of elements of different kinds. So some of those elements are genes. My old Cambridge boss, Willem Auerhand, would liken them to lights in a room, and other elements are more akin to regulators or dimmers for those lights. And I think the genes are certainly the most important of the elements, and they come in sort of two broad flavors. So you have coding genes and the non-coding genes. And there are about 20,000 coding genes, which is the stretch of genome that is transcribed into RNA molecules, which are then translated into proteins. So in the case of coding genes, the RNA's purpose is really only to mediate the production of proteins from that gene. But there are also tens of thousands of non-coding genes, and these are transcribed into RNA, but it stops there. The RNA is not translated into a protein. My research group specializes in the development and the application of statistical methods to discover the genetic changes or variants in human genomes that are responsible for rare diseases. And these disease-causing changes, they might happen in regions of the genome that previously would have been considered to be junk DNA.
And given that your area of research is based around rare diseases, what do you think these areas of non-coding genes, that's what we should probably refer to them as, that you looked at are actually responsible for?
So our publication in May in Nature Medicine concerns a non-coding gene called RNU42. We found that genetic variants in this gene are responsible for a neurodevelopmental disorder that we estimate affect tens of thousands of people around the world. In 2021, Daniel Green and my group completed an initial genetic association study of all the genes, so both coding and non-coding, across all the rare diseases in this very impressive Genomics England data set. We identified hundreds of associations with coding genes and also a few very promising associations with non-coding genes. And so we decided to write up a paper on the coding genes first, which appeared in Nature Medicine last year, before moving to the non-coding genes. And when we returned to our analysis of non-coding genes, we immediately focused on this really small non-coding gene called RNU42. This very small non-coding gene, it's only 141 bases long, contains variants which are responsible for this neurodevelopmental disorder, which is now called RNU42 syndrome or Renew syndrome. The syndrome features intellectual disability and other traits such as short stature, small head, seizures. And what's really striking about this is that we estimate it affects about 1 in 20 to 25,000 young people. And this would make it amongst the most common monogenic causes of neurodevelopmental disorders known to date.
How could, though, the mutation of a non-coding gene lead to the formation of a neurodevelopmental disorder?
That's a really good question. The RNA that this gene encodes is called U4, and it's one of the 5 small RNA components of a molecular complex called a spliceosome. So spliceosomes, as their name suggests, play a crucial role in splicing exons together within the nucleus. So exons are like linear islands in an archipelago. They are separated regions of genome which are transcribed into RNA, and then they're spliced together to form a mature RNA molecule, which can then be translated into a protein. So there are 2 types of spliceosome in humans. There's the major spliceosome and there's a minor spliceosome. And in fact, defects in 2 small non-coding RNAs, which are in the minor spliceosome, have been previously tied to a rare disease. Now, these RNU4-2 mutations that we published earlier this year, they represent the first reported non-coding defect in the major spliceosome.
So just so I've got this straight, the idea that these non-coding genes are responsible for splicing coding genes, but they may cut it at the wrong place, and therefore the recombination leads to, say, an incorrect protein, which could lead to a neurodevelopmental disorder.
That's exactly it. That's the running hypothesis. We just haven't really been able to find a large-scale defect in this splicing process in mature cells that we've studied from patients. And that's why this kind of lab work, where you take cells and differentiate them into different types of cells that we can't study from patients, and look at the splicing in those cells, could shed some light into where the splicing problems occur and which cell types does aberrant splicing occur.
It seems extraordinary that we've spent so long ignoring this huge other part of our genome. And I guess the mind boggles at how many other neurodevelopmental but also physical disorders could be down to errors going on in non-coding DNA that we've previously ignored.
That's right. I mean, cost of sequencing was such that there was a case to be made for only sequencing the protein coding genes initially. If you can sequence 50 patients instead of one by restricting the regions of the genome that you sequence to the protein coding genes, then that might initially lead to more scientific findings than if you sequence the entire genome, because it's reasonable to suspect that many, if not most, of the variants that cause diseases are located in protein coding genes. But by the late 2010s, the cost of sequencing a whole genome had gone down quite drastically. And so that made it much more attractive to sequence entire genomes in large numbers of patients. And that is what's allowed us to discover that indeed some non-coding genes contain mutations that can cause really quite severe diseases in humans.
I'm envisaging a future where this sort of research, looking more and more into non-coding DNA, could go hand in hand with better, earlier diagnoses of neurodevelopmental disorders, such as the ones you found, perhaps autism. Do you see that as being a potential future?
Absolutely. One of the things that has happened as a consequence of this discovery is that all the big clinical genetic testing labs have scrambled to adapt their tests so that they can pick up sequence variants in this small gene. And one of the issues with testing that restricts to specific regions of the genome is that they need to be adapted regularly every time that there's a new discovery. If it's in the non-coding parts of the genome, then the test will probably need to be adapted.
Ernest Turo, thank you very much. So what else could be lurking in our non-coding DNA? Well, on a broader scale, perhaps an organism's ability to speciate, that is, to evolve into a new species. It could be heavily influenced by so-called satellite DNA. This is DNA that encompasses our coding DNA and gives it structure. The three-dimensional structure of a gene can heavily influence how it expresses itself. So how the satellite DNA acts around it surely has some say in how those genes behave, and even the rate at which they mutate, the backbone of speciation. To find out more, I've been speaking to MIT's Yukiko Yamashita. And since we've leaned so heavily on book and recipe metaphors so far, why stop now?
So if you think about coding DNA, you can imagine that that is a actual, contents information of a book. Those words that you enjoy in a book, that is a coding part. On the contrary, we barely pay attention to the binding margin of the book because it doesn't give us any information. However, those, the binding margin is critically important because if we don't have it, all those pages are completely jumbled up. After you read a page 1, you don't know where the page 2 is, and you end up with jumping to page 527, and then, the next thing you see might be page 18, that kind of things. So one function the satellite DNA might be playing is to function as a glue or binding margin of the pages of the book to bring all pages, or in other words, chromosome in this case, together into one place so that, it doesn't get separated from each other.
So if you had two organisms with different satellite DNA, how could that affect their speciation?
So you could imagine, the exact same story. Two publisher can publish those two stories. And then one publisher might decide to bundle those pages on the side. And then on the other hand, other publisher might decide to print a book in a way that everything is bundled on the top of the pages. The story is exactly the same. But if you mix up those pages before bundling, and then how can you decide how to bundle them into, pages in the right kind of order? But then just because, the formatting information is not the same, you cannot just form the correct functional book. That might contribute to some sort of dysfunctionality of the hybrid cells.
Yes, I suppose if you had half of the pages from one book which had binding on the side and half the pages with binding on the top, they wouldn't be able to fit together. They'd be basically two different books.
Exactly. Yeah. So that could actually explain, right? Even when protein coding genes are extremely similar to each other, the two kind of chromosome from two species might not necessarily function very well.
So do we think the changes in satellite DNA could come about due to random mutation, same as regular speciation?
I think probably satellite DNA mutate even faster because coding sequences is so much of a constraint so that there's a selection against the changes, right? So that means if you are talking about the book, if you change just one character in a word, the whole meaning could change. Zoom versus zoo would be very different. And then also, the probably Zoom will be very different from boom. And then, the next change, like bloom, is going to be completely different. But then if it's just a formatting information, the satellite DNA is just a formatting information. And then, for example, satellite DNA can be just AATAT, repeating so many times, only to signal, to put the glue here or that kind of things. Let's say you have 10,000 copies of AATAT, still probably it's gonna work completely okay. But then 1,000 copies mutated to AATSC. I think, that there's minimal impact to the species. So that allows satellite DNA to change much more than individual words as a contents information. So I think that's why, the satellite DNA or those kind of non-coding junk DNA tend to change a lot. And then, by the time you realize, your formatting information, like binding merge information might be very different from your cousin species or cousin populations.
If that is the case then, albeit completely inadvertently, perhaps unfairly, do we think then that certain organisms with certain structures of satellite DNA may be able to adapt quicker than others?
Oh, that's interesting possibility. It's possible, I feel. Some species might get lucky to get adaptable kind of satellite DNA. I don't know if species or any organisms would have that foresight. So let's go that direction because that is going to make us better and then more adaptable, in the face of changes. For that, I want to be very neutral. But with that said, yes, in the hindsight, certain or the certain changes that happen to your satellite DNA structure, junk DNA structure, might make particular species a little more fast evolving. That's possible.
Yukiko Yamashita, thank you very much. So there you have just a couple of examples of what junk DNA is responsible for and capable of. The next few decades of genetic research have a whole new spin on them now, and we await to see what else is hiding in the junk. But for now, to round off this month's Naked Genetics, it's time to explore another quirk of evolution. You join me in the tropical, near-coastal waters of the Pacific Ocean, more specifically, right at the surface. The pelagic zone of the ocean is one that sees immense turnover on a daily basis as the abundance of sunlight and tidal currents cause an unimaginable number of microorganisms to float through. Staying on the surface is a banquet in a very open ocean, providing you can do two things. One, you can be sustained by jellyfish and hydrozoa, and two, you have some means of staying at the surface. This group of unrelated organisms that occupy the niche of surface water, or just below it, are called newstons. They employ a range of tactics and adaptations. Most things can just inherently float, like the Portuguese man o' war. Pond skaters do so by not breaking the surface water tension with their legs. And then we come to the curious case of Janthina janthina, the violet sea snail. The violet sea snail is a beautiful organism, as snails go. A shell consisting of light purple shading on the spire, down to even deeper purple on the ventral side. It measures about 4 centimetres by 4 centimetres, a colossus in the Newston world. But looks only get you so far in the animal kingdom. It's still a snail, so it isn't exactly fleet of foot, singular, and it can't swim, which is suboptimal for an ocean dweller. So if you can't float naturally, you're going to have to make your own raft. At the surface of the ocean, alongside all that food and also a trillion billion bits of microplastic, are air bubbles, pockets of O2 that haven't yet burst and joined the wider atmosphere. And the violet sea snail makes use of these in a truly extraordinary way. By wiggling its foot to and fro, this snail produces mucins, slime-esque proteins that come out of epithelial tissues. These mucins are amphiphilic. That is a complicated word for saying they love water and fat, so they stick to the snail and water. But they'll also trap air. So, by lassoing air bubbles with its own goop, this snail can hang upside down on the surface of the sea and catch a meal as it floats on by. But what if someone were to find it? Surely hanging out at the surface whilst being unable to escape is just asking to be snatched up by a hungry mouth. Or did you think those purple shadings were just for vanity's sake? This snail is not just a pretty face, no, this is a remarkable case of reverse counter shading, which I guess could just be called shading. For those out of the loop, many marine organisms, ranging from great white sharks to penguins, have a white tummy and a dark back. The deep sea is dark and the surface is bright. Therefore, anyone looking at your white tummy from below won't see you against the surface. Vice versa, anyone looking at you from above won't see you against the dark of the sea floor. This is counter shading, simple but brilliant. So why does the snail have a dark tummy and a light back? Because it feeds upside down, meaning the colours have to be flipped. Reverse counter shading. I could go on about this wild seafaring snail, how it changes sex halfway through its life to accommodate its eggs, how it can raft in groups to make even bigger flotation platforms. But the take-home message is this: if anyone ever says that you aren't suitable for a role, kindly let them know that a humble snail is thriving perfectly well at the surface of the big blue sea. That's it for this month's edition of Naked Genetics. If you like the show and are in a giving mood as we approach the festive season, do consider donating to The Naked Scientist to continue allowing us to make shows just like this one. We've made it very easy and secure, nakedscientist.com/donate. Do join me again next time for further fun genetscapades. If you'd like to drop me a line, my email address is wilty@nakedscientist.com. I've been Will Tingle, thanks for listening, and until next time, stay safe and goodbye.